Hello! Today, we're going to dive into the chemistry of a very common sugar, lactose. By the end of this video, you will be able to first, identify the monosaccharide units that make up lactose. Second, understand the specific chemical bond that links these units together. And third, describe the three-dimensional structure and anomeric configuration of this important disaccharide. Let's begin. So, what exactly is lactose? Lactose is a disaccharide, which means it's a sugar made of two smaller, simpler sugar units, or monosaccharides. The name disaccharide tells us there are two sugar units linked together. It's famously known as milk sugar because it's the principal carbohydrate found in milk. In human milk, lactose makes up a significant portion, about 7.2%. In cow's milk, it's about 4.7%. This makes it a primary source of energy for infants. The bond in lactose is broken down by an enzyme called lactase in our small intestine, releasing the two monosaccharide units, which are then absorbed into the bloodstream. We'll explore this process and the importance of the lactase enzyme in a later video when we discuss lactose intolerance, but for now, let's focus on the molecule itself. Now, let's address our first objective, identifying the building blocks of lactose. As a disaccharide, it's composed of two monosaccharide units. These are D-glucose and D-galactose. Let's first look at the structure of glucose. It's a 6-carbon sugar or a hexose. In its cyclic form, it exists primarily as alpha-D-glucopyranose and beta-D-glucopyranose, with the beta anomer being the most common. We will be using the beta-D-glucose structure here. Notice the position of the hydroxyl group on carbon 1. In the beta anomer, this hydroxyl is in the equatorial position, meaning it points away from the plane of the ring. Next, let's examine galactose. Galactose is also a hexose and is structurally very similar to glucose. If you compare the structures of glucose and galactose, what do you notice? Galactose is a C₄ epimer of glucose. This means they are identical in every way, except for the configuration around carbon number 4. In glucose, the hydroxyl group on carbon 4 is in the equatorial position, but in galactose, it's in the axial position, meaning it points straight up. This small difference has profound effects on the molecule's properties. So, to recap our first objective, lactose is made of 1 unit of beta-D-galactose and 1 unit of beta-D-glucose. Now that we know the two components, our second objective is to understand how they are linked together. Monosaccharides are joined by a special type of covalent bond called a glycosidic bond. The bond forms through a condensation or dehydration reaction. A hydroxyl group from the anomeric carbon of one sugar reacts with a hydroxyl group on any carbon of another sugar. In the process, a molecule of water is removed. This results in an oxygen atom bridging the two sugar units. For lactose specifically, the glycosidic bond is formed between the anomeric carbon, C₁, of the galactose unit and carbon 4, C₄, of the glucose unit. This bond is therefore called a 1,4-glycosidic bond. So, we have a C₁-C₄ linkage. But there's another crucial piece of information about this bond, its configuration. This brings us to our third objective, which covers the three-dimensional structure and anomeric configuration of the glycosidic bond. Glycosidic bonds can be alpha or beta. This refers to the stereochemistry around the anomeric carbon, or C₁, of the first sugar unit. In our case, the galactose. Let's re-examine our beta-D-galactose. Remember, the hydroxyl group on C₁ is in the beta position, meaning it points up from the plane of the ring. When the bond to the second sugar forms and that C₁OH group is in this up orientation, we call the resulting bond a beta-glycosidic bond. Conversely, if the C₁OH group were in the alpha position, pointing down, the resulting bond would be an alpha-glycosidic bond. A common example of an alpha-glycosidic bond is the one found in table sugar, sucrose. So, combining what we've learned, the complete name for the linkage in lactose is a beta-1,4-glycosidic bond. This means a beta-configured bond is formed between carbon 1 of galactose and carbon 4 of glucose. Let's put it all together and build the complete structure of lactose. We'll start by placing our beta-D-galactose and beta-D-glucose side by side. Here we have beta-D-galactopyranose on the left and beta-D-glucopyranose on the right. Next, we'll form the beta-1,4-glycosidic bond. This involves a dehydration reaction between the C₁ hydroxyl of the galactose and the C₄ hydroxyl of the glucose. As we form the bond, a water molecule is eliminated. The resulting oxygen atom bridges the two rings. The beta configuration is preserved, as the bond from the galactose ring points up, just like the original C₁₂H. The galactose unit is now referred to as the galactosol residue. And there you have it, the full structure of lactose. It's important to note the anomeric carbon of the glucose unit at the end. It's still a reducing sugar because its anomeric carbon is not involved in a glycosidic bond and can undergo mutarotation to form its open-chain aldehyde. Let's quickly recap what we've learned today. We started by identifying the two monosaccharide units of lactose, beta-D-galactose and beta-D-glucose. Remember, galactose is a C4 epimer of glucose, meaning they differ in the configuration at carbon 4. Then, we examined the bond linking these two units, which is a beta-1,4-glycosidic bond. It's a beta bond because it originates from a beta-configured anomeric carbon on the galactose, and it's a 1,4 bond because it connects C1 of galactose to C4 of glucose. Finally, we assembled the complete structure, visualizing the beta-1,4 linkage bridging the galactosyl and glucose rings. We also noted that the glucose end remains reducing. So, we have successfully met our learning objectives. You can now identify the components of lactose, understand its unique bond, and visualize its structure. Thank you for watching. To dive deeper into this topic and other related concepts, consider subscribing to our channel and checking out our other chemistry lessons. If you have any questions or requests for future videos, please leave a comment below.